Let's get started. Skeletal muscle cells, here we are. Mechanism of excitation is based on the neuromuscular junction, which contains the neurotransmitter acetylcholine, which binds to nicotinic receptors. Electrical activity of muscle cells requires action potentials. The calcium sensor is troponin C in the cross-bridge cycle. Excitation-contraction coupling requires the presence of L-type calcium channels and ryanodine receptors. The L-type calcium channels are in the plasma membrane. They are voltage gated. The incoming action potential depolarizes the membrane potential, changes the configuration of the L-type calcium channel, which is connected to the ryanodine receptor. The change of the configuration of the L-type calcium channel leads to opening of ryanodine receptors. Calcium leaves the sarcoplasmic reticulum and moves into the cytosol. Consequently, the concentration of intercellular calcium goes up, which triggers the start of the cross-bridge cycle. Terminator of contraction is the breakdown of acetylcholine and reuptake of calcium into the sarcoplasmic reticulum. Duration of the twitch, 20 to 200 milliseconds. Regulation of force is by the frequency and recruitment principle. I'll discuss in the present part. Metabolism is both oxidative and glycolytic. Here are some structural features of the skeletal muscles. We have here the bone and the tendon directly connected to the bone. The tendons are elastic. So here we have elasticity both in series like the tendon, but also in parallel. Different types of connective tissues containing collagen among others. So we have here the muscle and the fascicles, and then we have the muscle fibers. And here are the myofibrils, actin and myosin inside the muscle fiber. Remember, I have discussed this in previous parts. So the tendon is elastic. Connective tissues are also elastic. So now we have here elasticity in parallel, but we also have tightening. So we have different parts that account for the elasticity of the skeletal muscle, which is, of course, required for the cross-bridge cycle. Here are some more structural features of skeletal muscle cells. In the previous part, we discussed the structural properties of cardiac muscle cells here. Here we discuss the skeletal muscle cells. I have some notes here. Skeletal muscle cells, single, very long, cylindrical, multinucleate with obvious striations, which you can see here. Several nuclei, and here are the striations. We distinguish between three types of fibers. Type 1, type 2b. Type 2a is almost like a hybrid. It's a combination between type 1 and type 2b. Type 1 is normally reddish because it contains large amounts of myoglobin bound to oxygen. Myoglobin bound to oxygen gives it the reddish color. Type 1, we have here the example of a long-distance runner. So they require oxidative metabolism. Okay? Slow. Oxidative, long time. Type 2b, like the sprinter, 100 meters, 200 meters. Okay? This is the glycolytic type. This is normally whitish. Glycolytic mechanism. And then you have type 2a, which is a combination between type 1, type 2b, almost like a combination between the two. Here are some differences between the three types. I'll just quickly go over the list. Speed of contraction, type 1, remember this is the one that requires oxygen, okay, oxidative metabolism, so this is slow. Type 2b is fast. So, the myosin ATPase activity of type 1 is slow, type 2b is fast. This is the sprinter, remember. So, primary pathway for ATP production in type 1, remember, aerobic. This is the long-distance runner. Requires oxygen. Type 2b, anaerobic glycolysis, and the mixture, remember, hybrid, type 2A, aerobic, some anaerobic glycolysis also. Myoglobin content of type 1 is high, type 2B low. Recruitment order. Type 1 first, type 2A second, type 2B third. Then, rate of fatigue. Type 1, the long distance runner, remember, slow fatigue, fatigue resistant. Type 2A, intermediate, moderately fatigue resistant. And type 2B, fast glycolytic fibers, remember, fast, fatigable. The action potentials, remember I discussed it in this part. This is the action potential in the skeletal muscle. Remember, I also discussed there is no hyperpolarization normally because the resting membrane potential is very close to potassium equilibrium already. Mechanism of excitation, well, we have here somatic control. An example is the knee-jerk reflex. I will discuss this in a separate part, but now just quickly go over it. We have here the tendon, okay? Then we have the reflex hammer. We tap the tendon here, the ligament. So then we activate this neuron, a sensory neuron. This sensory neuron goes to the spinal cord via the dorsal root ganglion. Then it synapses on a motor neuron, here, the red one, which goes back to the muscle and activates. So here we have, by the motor neuron, activated this muscle here, which is the quadriceps. So we activated with the motor neuron the extensor. At the same time, we have here an inhibitory interneuron. Now this synapses on another motor neuron here. Now this inhibits the hamstring, which is the flexor. Control skeletal muscle tension. We have two principles. Recruitment, which is based on recruitment of more and more motor units, or increase of frequency. Remember, this is one motor unit. I discussed this in a previous part. This is a motor neuron, neuromuscular end plate, and a muscle fiber or more. So we have an action potential followed by a twitch. If we increase the frequency of the twitches, we have tetanus. Here we have single muscle twitch, like 5 Hz frequency, then a second twitch. Now we increase the frequency to 10 Hz. We have more and more twitches. Even further up, 25 Hz. More and more twitches. This is called unfused tetanus. And eventually we have a fused tetanus. At 50 Hz, for example, here, you can see this behavior. Excitation, contraction, coupling, skeletal muscles. I have discussed this in details in these two parts. Quick recap. You have here the neuromuscular end plate. You have the neurotransmitter stored in the vesicles. Acetylcholine is the neurotransmitter. It is released. It binds to nicotinic receptors here. And eventually we will have an action potential here. So the action potential arrives here at the T-tubules, activates the L-type calcium channels, remember the plasma membrane. These channels are connected to ryanodine receptors in the sarcoplasmic reticulum. Opening of the ryanodine channels releases calcium from the sarcoplasmic reticulum, which kickstarts the cross-bridge cycle, remember. Here again, the videos 5 and 10 in my playlist. So now let's discuss the sources of calcium in the skeletal muscle. Remember, in the cardiac muscle, we had two sources, extracellular and sarcoplasmic. In a skeletal muscle, we only have one source, which is the sarcoplasmic reticulum. So here we have the action potential coming in. It depolarizes the membrane potential, changes the configuration of the voltage-gated calcium channels, the L-type, remember, So these calcium channels are connected to the ryanodine receptors in the sarcoplasmic reticulum. Change in the configuration of the L-type calcium channel here results in opening of the ryanodine receptors, which in turn is followed by a release of calcium from the sarcoplasmic reticulum into the cytosol. This is the only source of calcium in skeletal muscles. Remember, we discussed this in this video, synaptic transmission at the neuromuscular junction. So here, I would like to show you, to remind you again, the calcium sensor is troponin C, which I have discussed in this video. Now, determination of the contraction. We have two mechanisms. One is the breakdown of acetylcholine by the acetylcholine esterase, which is here. It breaks it down to acetyl and choline. Then we have choline reuptake here, which is sodium-driven. Sodium-driven co-transport for choline reuptake. Choline reuptake here reconnects with acetyl, so we have then acetylcholine again, which is pumped into the vesicles to be stored again. Calcium reuptake, remember, I've discussed this here in the cardiac muscle video. We need this pump, CERCA, in the sarcoplasmic reticulum. Pumps calcium back into the store. Remember, in the cardiac muscle, we also have this exchanger. In skeletal muscle, since the source of calcium is the sarcoplasmic reticulum only, we need the CERCA. I hope this was helpful.